Thank you very much. Merci et bienvenue ce matin. On va avoir ce matin un genre d'expresso, d'impression de ce qui se passe dans l'environnement, la biodiversité. So for the panel today, I'm going to change my name and I'm going to pretend I'm Larry Page, co-founder of Google. He's my cousin. I've got $1 billion in my pocket and I'm in the elevator going down I'm on my way to buy Facebook. I've got $1 billion extra dollars and I'm going to ask these panelists to give me one slide in 3 minutes as to why I should invest in their field. So that is a Herculean task that the human genome is nothing compared to the complexity of achieving what we are about to attempt. And over there to the left is the equivalent of the Stanley Cup. This is the Genome Cup. And the best speaker will walk around with that over lunch, and then we'll have to return it to Cindy Bell, to the official treasury of the cup. As you probably know, Genome Canada in the early days distinguished itself, I think, relative to other national genomics bodies in doing an integrated program. So we were funding human health, and we heard yesterday about everything that's been done, or many of the wonderful things that have been done in human health, but also Genome Canada early on invested heavily in plants, fisheries, applications, fisheries, microbial genome, environmental issues, and as well in the gels or ELSI field, and integrating the gels components into all of our programs, an integral part of our scientific programs, and also as a standalone activity. So we're going to get a cross-section of all of that. And if I could have the first slide, it's just a little reminder that this panel is going to be addressing issues of a consequence to all of us. There's no shortage of problems, so this group is going to be very busy. And I think we'll be surprised at how well this field has evolved. It was a bit of a sleeper in that when you meet somebody on the street, Genome Canada, human genome is the first idea that comes to mind. And the genomics technologies that were spread to the health research community more rapidly, perhaps, but made their way, and our colleagues took up these instruments and these technologies and these techniques, a bit of a sleeper side, and actually I think are coming out much more powerfully than any of us had anticipated. So we'll get little snippets this morning, and the first one to go, the first brave soul, is Sally Aitken, who's the Director of Forest Conservation Genetics at UBC. And Sally, you have 3 minutes.
So on this panel, we have the largest to the smallest organisms on Earth, which is quite fitting. The world's forests are becoming mismatched to their environments due to climate change. We're seeing those effects in terms of increased mortality and damage due to drought, heat stress, insects, and diseases. Our forests in Canada are tremendously important to our nation in terms of providing wood, fuel, fiber, but also providing habitat for other organisms and for sequestering carbon. So what can we do about this mismatch between populations and environments? Well, using traditional methods, we would put out field trials and evaluate population differences over decades, but we don't have that kind of time. So we are now using genomic tools to, first of all, understand how trees are adapted to their temperature and moisture regimes, and what the genetic architecture of key traits involved in adaptation to climate is. And then we can take that information forward and we can derive recommendations for seed transfer. That is, what trees should we plant where to withstand the projected changes in climate over the next 80 or 100 years. Now, the value of this lies in many areas. We plant over 500 million trees a year in Canada, and those trees are expected to grow and produce all of those resources we require, as well as ecosystem services from our habitats. It's a relatively small cost to reallocate what we plant where. We already have armies of tree planters, we have nurseries that grow seedlings, and we have systems for producing or collecting seed. Now, in terms of implementing a climate-based seed transfer solution, what we need to do is address public policy around what you can plant where. Most of Canada's productive forest lands are publicly owned, and they are covered then by a set of forest policies. And we need to evaluate and interact with stakeholders, with professional foresters, and with people in forest-dependent communities to come to solutions that will ensure that forests are diverse, resilient, and continue to produce resources and sequester carbon.
Very good. Thank you very much. That was forestry. I noted 500 million trees per year. We'll come back to that. Our next speaker, Elizabeth Edwards from the University of Toronto. We just heard her earlier. You should know that she is looking very regal as she sits there because, of course, she was inducted into the Royal Society last week. So congratulations to her. And that's why she has this.
Way to go.
Yeah. I think she's not a monarchist or something. To Elizabeth.
So you heard already what I talked about, and I think groundwater is a huge freshwater resource for the planet. And if we want to extend the issues that I mentioned earlier, we need to protect our groundwater and our freshwater resources, and we need to find better ways to treat wastewater and industrial process water, recycle water in our industries. Because the industries that we use in Canada, mining, pulp and paper, use tremendous amounts of water and degrade that water in the use. So I think that biological processes can have a huge role in recycling water, and particularly these anaerobic processes that have been understudied. So methanogenesis and anaerobic digestion is one that could be used a whole lot more in North America. Not only do you degrade organic wastes, but that you produce methane and energy to offset some of the costs. So I think the challenge for us in North America is to bring these technologies that are known but not embraced in North America for a variety of reasons, to bring them more into commonplace use. Thank you.
Very good.
All right.
That's why she got the award. This is great. Okay, so next we have Gerritt Voordouw, who at the University of Calgary, NSERC chair in petroleum microbiology. He has a totally uncontroversial subject, which he will elucidate in a minute.
And I think we're in a time surplus situation.
Yes, there we go.
Anyway, welcome everybody. Good morning. We've already heard, and we are all aware of the huge problems that the tremendous energy use of the world causes. I mean, we all know about global warming, and we all know about the role of fossil fuels in that, and it would definitely be much better if we had a much larger fraction of our fuels to be sustainable and renewable. But truth be told, we're not there by any stretch of the imagination, and by last count, minimally about 70% of the energy use of the planet is fossil fuels, coal, gas, and oil. So we really have to all think about what can we do best, and of course we can blame the oil companies, but really I think when we all look in the mirror and we think about the energy use that we all indulge in, I think part of the problem is really right there. So one of the things we can do, the use is hard to do for any particular group. I think that's a very much a political decision. But we can, for instance, work on improved extraction and minimal footprint. And if you think about the oil sands or the tar sands, whatever you like to say, for instance, we all know the pretty horrible pictures of tailings ponds. One of the things that goes on in tailings ponds is lots of microbial life producing methane from residual oil. Really, I mean, microbes can make a very positive contribution there in making, for instance, sure that this methane doesn't escape as a greenhouse gas. So essentially then, there's a fantastic opportunities for genomics. The subsurface has a colleague of mine, Steve Leiter, he likes nice analogs. He likened it the Amazon below our feet. And we really have very little knowledge of it because different from the oceans, it's very hard to sample. I mean, you typically have to drill, and that's very expensive. But nevertheless, as part of this project, we have been able to collect approximately 2,000 samples over the past 3 years donated by oil and gas companies, and we've done pyrosequencing and metagenomic analysis on those. So what has that really yielded? I mean, have we learned anything? So I've already mentioned, In tailings ponds, we know that methane is being generated at depth, but at the surface, it's actually being very effectively oxidized, so that the annual or the daily emissions are still of the order of 10,000 cubic meters per day, but the real production is 2 or 3-fold higher. So if we can somehow make these methanotrophs in the top meter of water more effective, then we could reduce emissions to zero. Likewise, in pipelines, there's some interesting management practices, but with metagenomics, we've been able to study the types of bacteria involved in corrosion, and we think we can manage pipeline corrosion better that way. So what are hurdles in implementation? I think it's fair to say that in downtown Calgary, there's not too many engineers knowing about biology first and metagenomics second. So that's a problem. However, Shell, BP, ConocoPhillips are all starting to hire microbiologists and bioinformaticians. It's a small number, but they get hired for the expertise they bring, not to retool. And so I think, and I should mention Suncor in that respect too. So I think I see a bit of a shifting tide and good prospects for this area in the future.
Thanks, Gerritt. That's very good. Thank you very much. So we've had 500 million plants, we have water, we have the oil sands. We're doing all right. 3 speakers in 10 minutes. Our next speaker, Vincent Martin, is the CRC chair in microbial genomics and engineering at Concordia. In his former life, he spent a lot of time at Berkeley working in biofuels, where I think not his lab mate, but somebody down the street was Stephen Chu, who's now the Secretary of Energy in the United States. So Vincent is very chirpy and happy right now because he's been hearing synthetic biology in every major talk we've had since the start, and I believe that will be his subject.
Right. So I think I'm nicely framed here between Gerritt and Curtis in that my goal is to eliminate his need using his information. Of course, this is not going to happen in the near future. I agree with Gerritt that oil is there and it's there to stay. But I think that we have to think seriously about alternatives and alternatives in the processes that we can develop to produce molecules that are originally pulled out of the ground from the hydrocarbons. Most people always think about biofuels, but if you take a gallon of crude oil out of the ground, or syngas for that matter, you can produce a lot of other things than fuels. The chairs that you're sitting on are probably made of nylon, which is made up of fuel derivatives. So we have to think about developing technologies that are providing alternatives that are going to be more sustainable in the long term, because the path that we're on right now is not sustainable. So what's the solution, and how is genomics contributing to that? If you look at microbes, they've evolved over the years, over the many years, to produce all sorts of things. And we're actually using them already to make useful things like antibiotics or ethanol. And everyone talks about ethanol. It's at the forefront right now because microbes make it naturally. We have about 16,000 genomic sequences of microbes in GenBank. It provides a huge amount of information and diversity, and really the blueprint to build custom-made microbes or custom-made microbial system to replace those molecules and those processes that are currently used in the petrochemical industry. So it's not just the blueprint of the organisms that we can use to build or improve upon, but it's also all the different parts, which could be the genes or the regulatory elements that you're going to need to build these things. Excuse me, that's also provided by the genomics information that's coming out. Of course, it's not all pie in the sky. It's already happening. We heard about Stephen's talk this morning, and there's many biotech companies that are aiming at doing this. There's major significant hurdles. None of them are insurmountable. And as time goes by, and again, we saw this morning, petroleum is only going to get more expensive, and the technology is only going to get better. And at one point, those paths are going to cross. We have to be ready to take advantage of those technologies when we are there in the time frame. And a lot of the technologies that we're talking about, it's pretty easy to engineer an organism to make, let's say, adipic acid that's used in making nylon. It's much harder to make that organism produce grams per liter on a scale of millions of liters. And a lot of the hurdles that we're facing right now, it's all about scale and how fast you can do this. There's hundreds of molecules that come out of the ground. Every time you want to make a new molecule, you have to engineer a new microbe. That's a tough process. It's a long process. Genomics can contribute to that in a really significant way.
Oh, there we go. Merci, Martin. And Vincent, you should know, for those of you interested in synthetic biology, Vincent has got a little working group, a discussion group, I think, in trying to work up a strategy and approach. So you're welcome to connect into that after. I think he'll be coordinating. Our next speaker did speak, Curtis Suttle, Professor of Earth and Ocean Sciences at UBC. Professor Suttle will have a few more words to add to his subject.
Thanks very much. I think the Google model is perfect because that, kind of a sketchy business model, turning it into one of the largest companies or most valuable companies on the planet. And I think the thing that's remarkable and why there's synergies here is because it's all about collecting information and what you do with that information, pulling it together. You need vast resources in terms of computational power and actually trying to collect that information together and extract information from it. So if we look at where we are right now, so we're sitting in a situation in which there's more microbes in a liter of water than there is people on the Earth, and yet almost all of that genetic information there has not been seen before and is completely unknown. And so the big challenge is here, so the promise is that ultimately that genetic information is going to be the source of all kinds of new innovation. For example, there's a wonderful paper that I look at that looks at networks, for example, and you can go and look at highly networked proteins which you find in different environmental samples, and if you find one of those proteins, you'll find another 10 proteins. Just like if you find one protein in photosynthesis, you can bet your house that you'll find another 9 proteins, because anything that does photosynthesis needs that. When we look at the metagenomic data, you find the same thing. You'll find highly networked clusters of proteins, but not one of those proteins is known. So that tells you that there's whole biochemical, biosynthetic pathways in these environmental communities of which their function is completely known. So the promise is that there's tremendous amounts of information that will dictate all kinds of new applications and insights we don't even know existed. The challenge is, and again, it comes down to information, we don't know how to do that yet. I mean, drug companies for years have been trying to look at the promise of all this diversity and how do we figure out what it does. And so you can take gene sequences, you can clone them into microbes, and you can produce proteins, for God's sakes, of which you have absolutely no idea what they do because they're part of a pathway or they're part of you don't know what the substrates are that are necessary. So we really don't know what to do with that information. And so ultimately, though that's going to require a large amount of capital, a large investment of resources, both intellectual and monetary, to try and actually tease apart that information. But ultimately, it comes down to collecting all that information and then trying to interrogate it in intelligent ways.
Fantastic. Well, we are a country with 3 major oceans. We are surrounded by oceans, and I think your discussion this morning has helped us think a little bit more about what we have to deal with and think about. Our last speaker, Steve Yearley, comes all the way from Glasgow, and at the party last night, I inquired as to whether he was Rebus's younger brother. For those of you who read crime novels, he confirmed that he's not Rebus's younger brother. However, like Rebus, he seems to be drawn to complicated issues. So his field is the study of policy and controversies. He's the director of the Genomics Forum at the university. And I was just looking at a few of the items, GMO foods and food safety, genetics and security, genetics and access to healthcare. So he's obviously perfect for this panel. I suspect he has a few observations and a few comments. And Steve, first of all, thank you for coming to our event, and over to you.
Thank you very much indeed. I'm actually from Edinburgh, like Rebus himself, and I live just around the corner from his fictional detective. I guess I was chosen to be here as the only Brit who would understand the Stanley Cup joke, having been to the Hockey Hall of Fame on my last visit to Genome in Canada. So I'm really interested in winning the cup today. Okay, so we've had a lot about the power and the promise, and being a kind of contrary social science guy, I'm going to talk about the promise and the power. So I think what's been very important this morning, has been the kind of the sense of optimism that pervades the kind of genomic technologies applied to environmental issues. So, I mean, I think one of the key things, particularly around climate change, is that there's a sense of it's true, but it's kind of hopeless. We don't know what to do about it. It's an insurmountable kind of problem. And a lot of what we've heard this morning suggests that it isn't, I mean, insurmountable. It's possibly all too surmountable if we just organize ourselves correctly about it. So I think one of the most important things about the promise is the sense in which the most daunting societal problems begin to look less daunting because we have some plausible looking tools for sorting those things out. And my first image here is obviously a slide about, it's also about energy use, but it's also about information and connectivity in Europe. And I think the other thing that comes out of the environmental metagenomics work that's very important from the kind of promise side is the sense that metagenomics and remote monitoring and so on give a new way of us understanding and checking what's going on in the environment in places that it's difficult for humans to reach or difficult for humans to have constantly staffed bases in. So I think there's a lot about the promise there. The power, however, is something that I think we haven't talked really quite enough about in this meeting. So Curtis mentioned about the questions about the patenting and who has control of the life that's in the oceans. But I do think there's a very strong issue about power and knowledge and power and patenting and power and control in these issues. We heard yesterday in Bart Knöpper's presentation a bit about the role of participation in technology governance. And I think one of the key things that's come out of the controversies around GMOs and to some extent controversies around synthetic biology is that we need to be smarter about public engagement with these technologies, about securing not just a kind of a marketing campaign that persuades people that this is great and we know what we're doing, but a genuine attempt to engage and find collaborative solutions from these kind of things. And so it seems to me, and here's my other slide, this is the thing that's beloved of social scientists of a certain ilk. This is the panopticon, a kind of an idea of what an ideal prison establishment would look like from the 18th century, where you could have one person who could watch everybody in the cells. They couldn't obviously watch them all the time, but the thing was that the prisoners wouldn't ever know whether they were being watched or not. And there's that kind of management ideal that in some sense, these tools give us a way to control what's going on in the environment. But if we attempt to centralize that and we don't think about ways of collaborative, participatory things, then there's a danger that these solutions will lose their public credibility and we'll have all those contests again. So participation is very important. Thank you.
Thank you. Sorry. Thank you, Steve. That's great. So we're in good time. We're going to have a little bit of a discussion on the panel right now, but I'd invite those of you who have questions to start coming up, and we'll open that up for a Q&A session in a few minutes. Now, I had a few, as I listened to the presentations going back, if I could start with Sally. I heard you mention that I think replanting, it was 500 million trees per year in Canada, and most of the replanting is done by the provinces who own the lands. Do you have any idea of the scale of the replanting effort, whether nationally or in BC? How much does it cost on a yearly basis? Order of magnitude? Sounds like a lot of trees.
Well, it costs about, I have to reach on this, but it costs in the order of $1 to produce a seedling.
Okay.
And it probably costs a couple of times that to get it in the ground somewhere.
Okay.
And so if you multiply that by that, at well over 500 million seedlings, there is a big investment there.
Yeah.
And that investment is being made now because there is an obligation to reforest areas. So that money is being paid by, usually by companies that hold the tenures and have cut the trees on those lands.
Okay. So in terms of, it's kind of like healthcare, right? I guess it's a yearly expenditure. Sorry.
Could I add something to that?
Yes.
Though I think more than the cost, it's not about the cost of the planting, it's about the value of the resource at the end of the rotation.
Yeah.
So we did some very modest economic projections that suggested that just for Alberta and British Columbia, the losses in growth due to maladaptation, if we keep just planting local material, would be in the order of maybe $500 million a year in terms of wood products alone.
Wood product, in just BC and
BC and Alberta.
Okay. Yeah. We're very familiar. We've got a similar dynamic on the East Coast, Quebec, and you're right. I think getting the trees that have the right properties or right adaptive properties or even the right physical properties for industrial processes. Second, Gerritt, your question over to you. You talked. You're in the subject which is in the news every day. Sure. I was in the embassy in Washington. That's all we did all the time. And of course, one of the issues was the last kind of big environmental debate about oil sands oil, crude going through the pipelines, and whether or not it is more corrosive. Right. And that's still kind of churning around. And I wondered if your own research speaks to that, or
well, so if I have to address that, I mean that has been investigated quite extensively, and the answer really is that yes, maybe it is somewhat more corrosive, but it's all manageable. In fact, the people I listened to said it's not more corrosive; it's about the same as heavy oil. So, so in short, there doesn't seem to be a problem. We have to accept that we can't have no spills. I mean, a nice analogy is the fidelity of DNA polymerase. I mean, you can make it instead of 1 in 10 to the 10th, you could make it 1 in 10 to the 13th, but it would cost you big time. So we're just not prepared to pay the amount of money. I mean, the product would be hugely more expensive if we forced every pipeline to be 3 times as thick, et cetera. So this does not go on. So a certain number of spills will occur. If you have brand new pipelines like they're planning to, the Keystone and the one they plan to do to BC, if that ever happens, really, the first 50 years I'd be really surprised if you had any problems with that. I mean, the main problem with pipelines is aging infrastructure. I mean, they're around for a long time. Some of them are 50 years old. They should really be replaced.
Now, I heard you speaking earlier on the use of microbial agents to, I guess, do maintenance on the pipeline. Can you go on?
Well, I mean, as part of this Genome Canada-funded project, I actually didn't look at my slide, but I hope the title of that project was up there. And hopefully it was in green. I'm red-green colorblind. I may have selected brown for all I care. But